Welcome to The Explainer. Today we are diving into one of the most fundamental processes in all of biology, DNA replication. It's how the blueprint for every living thing, from a bacterium to you, makes a perfect copy of itself. And this isn't some rare event. It's happening trillions of times a day inside your own body.
So, let's start with the big question that baffled scientists for a while. Think about it. Before any of your cells can divide to, say, heal a cut or just help you grow, it has to duplicate its entire genetic manual. How on earth does it manage to do that with such incredible precision?
You know, when Watson and Crick first figured out the double helix structure of DNA, it was a massive breakthrough. Right away, they could see that the two-stranded design hinted at how it could be copied. But just because you have a hint doesn't mean you have the whole story. So scientists got to work and came up with three main ideas for how it could happen.
Okay, so here were the three main suspects in the lineup: the semi-conservative model, the conservative model, and the dispersive model. Each one painted a really different picture of what the new DNA would look like after being copied. Let's quickly break down what each of these theories was proposing. The conservative model was pretty straightforward. You'd keep the original DNA molecule totally intact, and a brand new one would be built from scratch. The dispersive model was a bit wilder. It suggested the new DNA would be this patchwork quilt of old and new pieces all mixed together. And then there was the semi-conservative idea, which predicted that each new DNA molecule would be a perfect hybrid, one old strand paired with one new strand.
So, we have our three theories. Which one was right? Well, thanks to some really elegant experiments, scientists figured it out. And it turns out, as it so often does, that nature's solution was both simple and brilliant.
And here it is, the winner, the semi-conservative model. This image shows it perfectly. You start with one parent DNA molecule. The two strands unzip, and then each of those original strands acts as a template to build a new partner. The end result? Two identical DNA molecules, and each one is made of half of the original parent. Half old, half new.
And that's exactly why we call it semi-conservative, right? It's not fully conservative, the original molecule isn't kept whole, but it's not thrown away either. It conserves one of the two original strands in every single new copy it makes. Pretty cool.
Okay, so we know what happens. Now let's get into the really good stuff, how it happens. This whole process isn't magic. It's carried out by an incredible team of tiny molecular machines we call enzymes. Let's meet the construction crew. All the action takes place right here, at something called the replication fork. This is that Y-shaped spot where the parent DNA is being unwound and the new strands are being built. You should really picture this as a super busy, perfectly coordinated construction site, with different enzymes buzzing around, each with a very specific job to do.
First on the job is DNA helicase. I like to call this one the unzipper. Its whole purpose is to move down the DNA ladder and break the little hydrogen bonds that hold the two strands together. It basically just pries that famous double helix apart so the code can be read.
And next up, we have the star of the show, DNA polymerase. This is the master builder. As soon as helicase opens up the strands, polymerase comes in, reads the sequence of bases on the parent strand, and grabs the right matching nucleotides to build a brand new complementary strand. It is absolutely essential.
But here's where things get a little tricky. You'd think you could just build both new strands in the same way, right? Well, not quite. The way these enzymes work creates this fascinating engineering problem that the cell has to solve on the fly.
And the whole problem comes down to this one simple rule. DNA polymerase is a one-way street. It can only add new nucleotides to one specific end of the DNA chain. Think of it like a train that can only move forward on its track. This little rule has huge consequences for how one of the new strands gets built.
Because of this one-way rule, one of the new strands, we call it the leading strand, is super easy to make. The polymerase can just cruise along in one long, continuous piece right behind the unzipping helicase. But the other strand, the lagging strand, runs in the opposite direction. That means it has to be built backwards, away from the replication fork, which is a real logistical headache. So how does the cell handle this? With a really clever workaround. First, an enzyme called primase lays down a little RNA primer, kind of like a starting block. Then, DNA polymerase jumps on and builds a short chunk of DNA backwards until it bumps into the previous piece. And it just keeps doing this over and over again. Primer, fragment, primer, fragment.
And these little short, disconnected pieces of DNA that get stitched together to make the lagging strand, they actually have a name. They're called Okazaki fragments, named after the scientists who discovered them. They're the signature of this whole lagging strand process.
All right, so you've got all these fragments. The last step is to connect them all into one solid piece. For that, the cell brings in an enzyme called DNA ligase, and its nickname is perfect. It's the molecular glue. Its whole job is to go along and seal the gaps between all those Okazaki fragments, creating one single unified DNA strand.
Whew, okay, that was a lot of molecular detail. But why does any of this matter? This incredibly complex dance isn't just a cool biology fact, it is fundamental to life itself. It's the reason you can grow from a single cell, and the reason a paper cut on your finger can heal.
At the end of the day, the final result of this entire, complex process is this. Where there was once one molecule of DNA, there are now two, and they are perfect, identical copies of the original genetic blueprint.
Now, this isn't happening randomly. The cell is incredibly smart about timing. DNA replication only happens during a specific window in the cell's life called the S phase or synthesis phase. This timing makes sure that by the time a cell is ready to divide, it has a complete extra set of instructions ready to pass on.
So let's just boil it down. Why is this so crucial? Well, it's the engine of growth and repair for every living thing. And even more profoundly, it's the mechanism that ensures our genetic information gets passed down faithfully from one generation to the next, keeping the story of life going.
And you know those classic X-shaped chromosomes you always see in biology textbooks? Well, this is where they come from. After replication, those two identical DNA copies get packed up super tightly. Each half of that X is called a sister chromatid, and they are perfect clones of each other, all set to be pulled apart when the cell divides. What's truly mind-blowing about all of this is the accuracy. That master builder, DNA polymerase, actually has a proofreading function. It checks its work as it goes, and if it makes a mistake, it can go back and fix it. The result is a copy that is astonishingly, almost perfectly, faithful to the original.
But that brings us to one last, really important question. I said it's nearly perfect. With trillions upon trillions of copies being made in your lifetime, even a tiny error rate means mistakes will happen. So, what happens when this incredible, precise machine makes a little typo? What are the consequences of just one tiny mistake in our genetic code?